Hello, everyone, and welcome to another Terra training module. I'm Frank Ambrosio, a bioinformatics scientist with Theogen Genomics, and in this video, we will be going over the process of uploading paired end Illumina sequencing data to Terra.bio. If you would like to follow along with today's exercise, please use the link in the description to download the example dataset. Before uploading data, one must register an account with Terra.bio and create a workspace. If you have not completed these steps yet, please take a look at the training modules on these topics before proceeding. If you have any trouble with this process, please reach out to our support email, which is support@terrapublichealth.zendesk.com, and we will include a link to that support email in the description of this video. Now that we have confirmed an account has been registered and a workspace has been created, let's navigate to the Terra upload page and get started. To navigate to the Terra upload page, please enter the URL app.terra.bio/#upload. Upon arriving at the data uploader, please select your workspace where you would like to upload this data. And you will be asked to either select or create a collection. So the collections within the workspace allow you to organize your data by sequencing technology. So for example, if you have sequences produced with Oxford Nanopore Technologies MinION sequencer, you will create a separate collection from your Illumina sequencing data. The workspaces themselves can be used to organize your data by organism. So you can have multiple different organism specific workspaces, and within those workspaces, multiple different sequencing technology specific collections. So in this example, we will create a new collection to upload our data to, and we will call it Illumina Training Collection. And you know what? Let's call it Illumina Paired End Training Collection in case we want to upload single end Illumina data as well. So then click Create Collection, and you can either drag and drop your files into this area, or you can use this Upload button in the bottom right-hand corner. For Illumina paired end data, you will have pairs of FASTQ files for each sample, and here we're uploading 10 samples, so 20 FASTQ files. And depending on the speed of your internet and the size of the files, it will take a couple of minutes to upload the data. In the meantime, let's take a look at our metadata sheet. So every time you upload sequencing data to Terra.bio, you'll be asked to include a metadata sheet. And this metadata sheet includes the sample names, the read files, and the run ID. Here, since we have paired end sequencing data, we have 2 columns of read files. If we only had 1 column of read file for single end data, that would be fine as well. But in this case, we have paired end data. So a special amount of attention should be given to cell 1A here. This is where we define our root entity. The syntax for defining your root entity is to write the word entity, colon, and have no spaces. Then you will define your root entity, and this will be the name of your Terra data table in Terra. And then your root entity must also be given this suffix _ID in the metadata file. And this is just how it is detected by the computer. So once you have your root entity defined, you will define the sample names themselves. You will provide the names of the files that you will be uploading, and then the run ID. And this run ID will come in handy later when we're trying to organize our data within Terra. So that's your metadata file. Let's take a look at our upload and see where the progress is on that. Okay, we have completed uploading all of our files. We can select next, and now we can upload our metadata files. Here you can drag and drop your metadata file into this area, or you can use the upload button. Here we have a metadata file ready to go. So we click on that, click upload. This workspace already includes a table with this name. That's fine. This is just an example. In your example, you should not see this error. Here we can download the files. So to ensure that the file names match between the files we've uploaded and the file names in our metadata file, we just need to make sure that we can click on these links and that there is the option to download. So once we have all of these samples uploaded and we have our metadata uploaded, we can click Update Table or Create Table in your case, and we'll be given this Done message. So our files have been successfully uploaded to our Terra data table, and we can check that out now in our workspace. So we can go to our workspaces page and select our workspace where we have uploaded our data. We can select the data tab within that workspace, and we should see our data included in a Terra data table here. We'll see all of our sample names, the read1 files, the read2 files, and our run IDs. So if we wanted to select only a portion of the samples, if we wanted to select only run 2 on this MySeq run, we can search by run ID and it will filter the data down just to the samples we want to see. We can also use this gear in the top right to select which columns we would like to see. We can sort them alphabetically. And if we wanted to download this information for any reason, we can click this download all rows button, which will download the entire Terra data table. If we only wanted to download the run 2, we can filter by run 2, select just those samples, and then click copy page to clipboard, and we can then paste this into an Excel spreadsheet. So that is how to upload Illumina paired end sequencing data to Terra.bio. Again, if you have any questions, please reach out to our support email at support@terrapublichealth.zendesk.com, and that link should be in the description of this video. So after this, you will probably want to analyze your Illumina paired end sequencing data. So go ahead and check out our subsequent videos on running the Titan Illumina PE workflow. Thank you very much, and good luck analyzing your data.